Hello, my name is Marina Yurieva and I'd like to present my work on the estimation of accuracy of genetic demultiplexing in mouse. Genetic demultiplexing is a technique designed to distinguish genetic identity of each cell in single-cell datasets from genetically diverse samples. Demultiplexing is a tool that uses genetic variation to identify cells of genetically diverse samples, separate them into singlets, and remove doublets. Demultiplexing has been used on multiple studies to separate human single-cell data, but it hasn't been shown how well it works on mouse data. Our goal is to test it on several mouse strains and estimate its accuracy on samples with lower number of reads and find how many reads can accurately detect the strains from each other. We also want to see if it identifies RNA single-cell data as well as ATAC data. What happens if we use single nucleus protocol instead of single cell? Does it identify data from different tissues and different strains with the same accuracy? At JAX, we have various lab strains and several well-derived strains which are more genetically diverse. We picked Black 6, NZO, and CAST as one of the most common lab strains, and CAST from the well-derived strains for our analysis. We used RNA-seq and ATAC-seq, and RNA-seq was sequenced using single-cell and single-nucleus methods. Also, we are sequencing both adipose tissue and pancreatic isolates, we have one sample for each strain and each condition, and for ATAC-seq data we have 2 replicates. We also noticed that our B6 sample got contaminated with CAST. It wasn't identified with with cell ranger 3, but was picked up with cell ranger 5. And first we wanted to remove it, but then we decided to keep this sample to see if downsampling strategy still picks up the contamination. This is our downsampling strategy. We mix cells in silico from different mouse strains with BAM merge, and then subsample randomly reads from different cells to 10, 5, 1, and so on %, and run Demultiplexing on this. We generated several pools of strain mixes. From isolate we are using 2 pools. We're not mixing B6 with CAST because it was already contaminated with CAST. For adipose we're also using 2 mixes of 2 and 3 strains, and for ATAC we're using 5 mixes. The accuracy of the data was more than 99%, even for the lowest ratio of downsampling. The number of correct calls increases with the ratio of downsampling, and the number of incorrect calls decreases. And we also notice a strange drop and then increase in accuracy in one of the ATAC samples. And overall in ATAC samples, we notice that in the original mixes, the ratio of correct calls is lower than when it gets downsampled. This is the opposite from what we expected, so we decided to investigate that. And we looked at the number of cells and number of reads during downsampling, and we can see that the number of reads drops significantly during downsampling, while the number of cells changes only slightly. And then we went back to our pure samples and looked at the number of cells per sample and number of reads per sample per cell in those samples, and noticed that CAST has the lowest number of cells and the highest number of mean reads per cell. And that was the sample that was in the mix that showed this strange curve. And we also think that maybe during our downsampling we are randomly picking reads, but it picks more reads from the cells with the higher number of reads, and those cells might have better quality and get identified better. So we decided to change our downsampling strategy and not to mix strains, and to get one cell and downsample the reads within each cell in those strains. So we have first we sample 16,000 reads and then divide that by 2 at each step, and the lowest step is 32 cells reads per cell. Now we can see that the number of singlets decreases in all the samples with the lower number of reads per cell, and number of doublets and ambiguous cells increases. We see a similar picture with the accuracy of correct calls. Overall, the accuracy of genetic background detection works with more than 99% accuracy, even in cells downsampled to 256 reads in ATAC data and about 1,000 reads in RNA-seq data. Doesn't seem to identify differently between adipose tissue and isolates in the sequencing strategy. Single cell and nucleus don't play significant role. Our previous data showed that Demultiplexing can identify only 86% of singlets in single-cell human data with about 28,000 reads per cell. In mouse data, it can accurately identify cells with much lower reads number per cell, and ATAC shows a higher accuracy than RNA-seq. We are in the process of publishing this workflow on GitHub. And I'd like to thank everybody who was participating in this project, taking care of mice, designing the experiments, and generating the data. Thank you for your attention.